Hello, everyone, and welcome to the Accelerating Progress in ICDA Year 2 section of our plenary meeting. And I'll be the moderator for this session in place of Rachel, who unfortunately could not be here today. So I'll try to do my best to moderate this great session. We have several speakers. We'll start with Eric Fauman, who will update us about the development of a gold standard SNP to gene dataset. That is one of the recommendations by ICDA. Then we'll hear about the advances in the field of polygenic risk scores by Cristen and Mike Inouye. And finally, we'll end up with Nicki Tiffin and Andrés Moreno-Estrada, who will update us about the developments of the Global Equity Working Group at ICDA. So without further ado, I will let Eric to get started with his the first talk.
Thank you for that introduction. I'm really excited to present this work that I've been doing as part of ICDA with the ICDA Working Group number 17 on generating a gold standard SNP to gene list. This was one of the original recommendations out of the ICDA white paper. The goals are to collect a set of gold standard to say for this locus, for this SNP, this is what we believe is the true causal gene. To do this in a way that is transparent and can be carried forward. And it's been really my pleasure to work with a super outstanding group of scientists, subject matter experts in all of these. And it's been a great experience. So as we think about what should be on this list of gold standard SNP to gene mapping, the gold standard genes, you really have to start with the question of what is the ground truth? Where do we begin? So one place to begin is with protein QTLs. If the trait is protein abundance, then a reasonable hypothesis is that the gene that encodes that protein, the cognate gene, is going to be the causal gene. And there have been a large number of really terrific wide pQTL studies that included lots of proteins and thousands of subjects. One recent one that I'm referencing here is from Peitzner et al. And colleagues, which was a soma scan in 10,000 people. And this study, like many before it, showed that there was a really strong distance dependence, that the cognate gene is usually very, very close to the protein, to the SNP for that protein. This is sort of derivative of that, showing that actually if you take the log of the distance, so to look at all intrachromosomal pQTLs, so those range in distance from 0 base pairs to 230 million base pairs. If you look across the whole Peitzner et al., you get actually 2 different peaks. And I don't have time to do this justice now, but the preprint of this just went up on bioRxiv this morning, and so you can read a lot more about it there. But the point of this is that there are clearly 2 regions. There's a closer in region of what we would interpret as cis-pQTLs, which is the red peak here. And then there's a region at larger distances, which corresponds to basically if you pick 2 random points on a chromosome. And these are 2 different biological phenomena, and they have a pretty clean separation at about 1 million base pairs. And so this is sort of the first handle we can get as we think about how do we establish what are true causal genes. And then there's a whole bunch of features that you see people use and reference when they start to describe what they think is probably the causal gene at a locus. And these are 4 that we as a team felt pretty confident in, being ortholog or mouse knockout information or mouse ortholog information, rare disease information that's coming from rare coding variants, and proteins that are known drug targets for the disease or related disease. And these are orthologous features that we tend to use, but they also are features that cluster close to GWAS SNPs. And so since we know that causal genes from the pQTL work tend to cluster at those GWAS SNPs, the fact that these other types of genes also clustered near GWAS SNPs gives greater confidence that these are in fact features that we can use to help annotate or identify the true causal genes. And just as one quick example, and I hope everybody's seen this paper from Regeneron from late last year. They really did a nice job of showing that the genes that have a burden for a particular trait, have a rare variant burden for a particular trait, are also clustered at these GWAS SNPs. So this is kind of confirmatory on both of those, that we are probably right in our intuition that a non-coding variant near a gene for which there is a coding variant is probably still the right gene for that trait. So to take a quick application of this, a quick game of who's that causal gene. This is a GWAS of hemoglobin concentration at a particular locus. And of course, like all loci, there are a lot of different genes that could be here. I posted this again to Twitter a few days ago, and I think within a few seconds, people responded with what is the correct causal gene, which in this case is EGLN1. And I'll explain that in a couple slides. But the point is that there are actually hundreds, and I'll show you about 1,000 loci where we have enough biological information where we can make a pretty good informed identification of what is the likely causal gene. So for those that don't know this locus or this gene, just really quickly, this EGLN1 is this really elegant intracellular molecular oxygen detection system where ordinarily HIF1 alpha, as it's produced, very quickly gets modified by EGLN1 and then quickly gets degraded. If you don't have oxygen around, EGLN1 can't perform its function. The HIF1 alpha goes on with its transcription factor activity and induces a bunch of proteins, including erythropoietin, which increases hemoglobin production and red blood cell production. So even though I haven't shown you here exactly how that particular variant is influencing EGLN1, it's highly likely that this is the responsible gene. And going further, as we look through the different features that one might consider, the mouse knockout of EGLN1 has an appropriate phenotype. There's a rare disease phenotype for loss of EGLN1. For some related traits, there is a missense mutation in EGLN1 that has the same phenotype. And in fact, there is a drug targeting EGLN1 for a related phenotype. And so what we've tried to do is, again, many of these stories, individual experts know for individual genes. If you were around early at the beginning of this whole thing, you heard the story about CARD9 and SLC39A8 for IBD. So there are lots of these where, you know, if you have studied that locus, you have a pretty good idea and explanation for what that causal gene is. In this case, what we're trying to do is capture this information, condense it down to a computable form. And so here, this is saying that EGLN1 can be the causal gene for these traits, which are now presented here as EFO terms. And then, you know, a human readable sentence of why that should be. And then what is sort of the equivalent of what the functional genetics work would be if we didn't already know what EGLN1 did. This paper that's referenced here kind of does the functional work, which would probably convince you that this is a very probable causal gene. In this case, as in many cases, the functional biology work was often done years or decades before the GWAS, but it still gives us the confidence that we've probably got the right gene. Now, one thing that one has to bear in mind constantly is the fact that it's very easy to build biological stories. This is a sentence that's kind of burned into my mind. The criterion of biological salience is surprisingly inclusive from this paper on schizophrenia candidate genes. And, you know, when you look at, if you say, okay, what mouse phenotype would be appropriate for saying that a gene is probably the right gene for one of these traits? Well, you can see EGLN1 actually belongs to a lot of different categories, or has a lot of mouse phenotypes, going from the very specific, which probably wouldn't apply to most genes that come up for red blood cell traits, to things that are probably too generic to be of any utility. So at the bottom, right, we've got this category of homeostasis genes, which is probably too broad. So really the question is what level of detail is most appropriate. And I'm going to say we don't really know at this point. We didn't try to solve that problem, but what we can do is be extremely transparent in what level of evidence we are using to support any of these annotations. And so here's an example where for this, we've picked this particular level of granularity, and you can see there below. Then again, this is in a computable form to say a GWAS of one of these EFO traits can be justified or rationalized on the basis of a gene falling into this mouse knockout category. So putting this on, again, on a very transparent footing. So at this point, we've collected about 1,000 of these, and you can see the breakdown here of from the metabolites to specific diseases like diabetes and IBD, bone mineral density, blood cell traits, a handful of trans pQTLs, and other traits like eye color and hair color and things where there are some very well-known causal genes. The assertion of saying this gene for this trait is a purely human endeavor, a member of the team looking through and saying, yes, we believe there's enough evidence for that. What I'm showing up here in beige is once that assertion is made, then through the other tables that we've generated, then these can be automatically annotated. So trying not to sort of put the cart before the horse, one hopes that once we've accumulated enough of these rules, we can build better methods that will do the automated selection of what the true causal gene is. In this case, we're saying, yes, we reviewed all the evidence. A human believes that this is the right one. Here's supporting evidence based on the rules that we've provided. What we have not done is built a table specifically of this SNP is causal for that trait because of this gene. What we've done is annotated loci. And here again, I showed EGLN1 is a likely causal gene for a number of blood cell traits. If you see a GWAS SNP in that region, an advantage of this is, I think it matches with the intuition that if we go to a completely new population and find some brand new SNP, which is now associated with hemoglobin concentration, EGLN1 is still probably the right gene, even though we haven't specifically annotated that one. What I'm showing here is if you go through the GWAS catalog and say, okay, what are all of the SNPs in the vicinity of EGLN1 that have a hemoglobin type trait, where are they? What I'm showing here is on the left, this color diagram is the p-value for that trait. On the right here is the distance of that SNP from EGLN1, as is often the case. There's a correlation here that as you get closer to the gene, the p-value across all these different traits actually gets better. So currently, or as of last week, there are about 240,000 entries in the GWAS catalog with a known position in GWAS genome-wide significance. And from these 1,000 gene traits like EGLN1 and CARD9 and MC1R and so forth, we can solve or annotate about 30,000 of them to say that we have a rule, we have an assertion that this particular gene is probably causal for that trait, and here's why. Here's the breakdown. A large number of them are cis pQTLs. There are some specifically annotated trans pQTLs, some metabolite QTLs, and then other, which is all the diseases and other traits. As I showed, EGLN1 by itself already highlights about 30 different entries in the GWAS catalog. Just as a quick sanity check or comparison to what we know from pQTLs, if you just for each gene pick what is the strongest annotation for that gene, which for EGLN1 would be one of those ones in the middle, we see that about 70% of the time that gene ends up being the closest gene to the GWAS SNP. And this breakdown of distance follows that we expect from what we observe from pQTLs and EQTLs and metabolite QTLs. So what we've generated and what we're working hard to get out to researchers as quickly as possible is these set of references, basically a set of what are these gene to trait rules, what are the criteria by which a particular mouse phenotype or rare disease trait can be linked to a particular trait, and then the application of those to the GWAS catalog. So I'm out of time here, but again, what we've aimed for, what I think we've achieved is a flexible and transparent framework for how we've annotated these, what we would like to believe are true causal genes that people can use to either annotate their own GWAS or begin to build models or train models as we try to get better at automating the process of mapping SNPs to genes. It's open and extendable in that we've mapped loci, not SNPs. We've annotated trait families according to the experimental factor ontology. And so as, for instance, if there were a new hemoglobin trait that's not currently specifically annotated, but it falls under it in the ontology, that would also be annotated. And I will stop there and take any questions. And thank you for your attention.
Okay, thank you, Eric, for that wonderful presentation. Take a look at Slido. Let's see. So far, we don't have any questions that I see on Slido, so I'll go ahead and ask the first question. So if I understand correctly, or better, can you tell us whether this tool, once it's fully developed and available to the whole community, how would it assist to interpret possible causality in a multi-ethnic framework where we are trying to maybe extrapolate associations found in different ethnic groups? Are these resources that are informing your algorithms independent of the genetic background of individuals, or they will be subjected to those problems as well, and we may need different versions of this tool for different ethnicities?
Well, that's a great question. So as I said with EGLN1, what's in the table is basically saying EGLN1 is a gene that in human biology is related to hemoglobin traits. And I guess I believe that that's going to be true regardless of background and ancestry. And I do think one of the advantages of annotating loci and not SNPs is, as I said, if you find a new population with a new SNP that associates with hemoglobin levels, near EGLN1, that should still be your first hypothesis, even though that particular SNP has not been observed. I think, I certainly, as I've looked at Biobank Japan and other resources, certainly genes overlap quite a bit, but I think the pathways then also do. So you do sometimes find the same pathway or same types of genes, even if they're not the exact genes. So the way I would imagine this being used is if you have a new GWAS on a trait in a new population, first you would run it through this and it would flag, here are all the loci that have sort of been well characterized before. But then, based on that, you would say, this trait seems to, have a lot of cartilage genes, or has a lot of genes in this KEG pathway or whatever, and then you would be able to look to see in other new loci, unannotated loci, if there are genes that look like the annotated loci. So to a first approximation, I think it would translate well from population to population. Of course, there are going to be gene by environment effects, and if those got really strong, so that completely different pathways are at play in different populations, then it might have more limited application. But.
Okay, great. We have now a few questions. So I'll start by the most voted one, by Anonymous. Are you doing this gene in a tissue agnostic or tissue specific way? Do you have any SNP that point to different genes in different tissues?
Yeah, that's something that's been brought up, certainly for version 1, it's tissue agnostic. I think there are some nice examples of loci that seem to have different causal genes in different tissues, and that I think would be a nice addition in version 2 to say, right, if you know that your SNP is acting in a particular tissue and this gene only has relevance if it's acting in that tissue, then that's the more likely one. Currently, there's no tissue information either on the GWAS side, the SNP side, or the gene side.
Right, but how is there EQTL data that is informing your decisions too? So
every time I've looked, EQTLs do not improve mapping of SNPs to genes.
Okay.
Right. And we have I'm privileged to be on 2 papers that have come out in the last couple months that sort of reiterate this. One is the open targets approach, and the other is the effector index. Both of those show that EQTL information, if it helps at all, is way down the list.
Wow. Okay, very interesting. From Adam Butterworth, an incredible amount of work to map those 1,000 genes and over 30,000 associations in a robust gold standard way. What would it take to increase that to a much higher fraction of all GWAS associations? Is it person power, crowdsourcing, better algorithms,
yep.
AI, ML?
Yeah. Well, all of the above, but yeah, certainly I'm envisioning, I would like to see this be crowdsourced. So that's why I've put a lot of time and effort into trying to put together a robust framework so that, right, I mean, I've looked at a lot of loci, but the sum of all geneticists is much larger, and I know there's a lot more expertise out there. So I'm happy to take other questions, reach out to me however you want. I want to make sure we have time for other speakers.
Yeah, probably we should move on to the next speaker. Thank you very much for this great presentation, very interesting work. And now we are going to move towards PRSs. And we are going to start with Cristen Miller, professor of internal medicine, human genetics, and computational biology from Michigan State University.
Hi, everyone. So we're going to talk for a few minutes about the ICDA Polygenic Risk Score Task Force. And in a couple of minutes, I'll hand it over to Mike Inouye, my co-chair of this, this task force, to describe some of the findings. And I will briefly introduce our group with thanks to Kate Balakonis for her organizational wizardry. So my role is really to introduce the genesis of our task force. That's my disclosure. And this is our fabulous team. So polygenic risk scores were first discussed at the ICDA organizing committee. And the question was, how do PRSs fit into the initial mechanism or sort of map we drew up between maps to mechanisms to medicine within the ICDA? And not only does polygenic risk scores not start with M, but also how does it fit in that sort of very traditional finding an association, mapping the gene, like Eric so nicely just described. Developing new medicines, polygenic risk scores is a little bit outside that, but clearly it's an important approach for improving human health by taking genetic information and prioritizing which individuals might best benefit from specific medicine. So together with co-chair Mike Inouye, we assembled an international team of experts in clinical, statistical, and ethical aspects of the development and deployment of polygenic scores in clinic, with the hope of providing a balanced perspective of the potential usage of polygenic scores towards the broader goals of ICDA, as well as the goals of the genetics community to improve human health in an equitable way. So to very briefly introduce polygenic scores, and we already heard about some of them earlier in the day, but basically we expect that many genetic variants across the genome and the genes that are influenced by those variants likely influence the common diseases that we study. We can therefore, once we have mapped these variants to their locations in the genomes and try to estimate their influence on disease, we can then for each individual come up with a single score to represent that individual's genetic risk. And on the right-hand side, you can see a distribution. And once we develop a single score for each person, we can identify individuals with particularly high risk of disease. Just briefly, people typically use these terms somewhat interchangeably: a genomic or genetic risk score, a polygenic risk score, which typically applies to a disease, since we're talking about risk. And some people use polygenic score or PGS to describe any disease or trait. Right now, they're all approximately interchangeable. So very briefly, how is a PRS constructed? We basically sum the risk alleles for a specific individual at a number of disease-associated markers. We weight each marker based on our estimate of the influence of that marker on disease risk. And the choice of which and how many markers we include in that model can differ widely across different diseases, different methodologies, different ancestry groups. Some people use genome-wide significant markers, maybe ranging from a handful of markers as high as full genome-wide with Bayesian adjustment of the weights. There can be machine learning models for marker selection. So keep in mind that the marker selection can vary from dozens up to millions of markers that are fed into that estimate. And then typically that estimate for each individual is converted into a percentile. So an individual can evaluate their polygenic score relative to others, typically in the same ancestry or demographic group. So to introduce the tasks that our task force was tackling, there's a tongue twister. Within the ICDA goals and recommendations, we were focused on recommendation 14, which was to broadly ensure responsible use of polygenic scores in medicine. And our sub-aims were to assess the major gaps and risks related to polygenic scores, to identify and enumerate best practices as they develop over time. We wanted to address issues related to applicability of PRS across and within ancestry, as well as other axes of diversity. And lastly, we wanted to recommend actions to ensure that polygenic scores are deployed responsibly and equitably, including transparent disclosure of the importance of polygenic scores, but also the uncertainty related to them. We wanted a diverse group of expertise within the task force, and I think the group that we came up with was beautifully suited for this task. So briefly, when we asked people to provide their academic expertise, we developed a word cloud. You can see on the left of the various academic expertise of all the different group members. And although we're not perhaps as global as we will be in the future, we did at least have some representation from major continental groups internationally. So with that, I'll pass you over to Mike Inouye to talk about the recommendations and findings from this group.
Wonderful. Many thanks, Cristen. So next slide, please. So what are the potential benefits, risks, and gaps for using PRSs in clinical practice? That was the question that we sought out to answer. And here, maybe one back. And here I should clarify that the PRS task force interpreted responsible use as where the benefits outweigh the risks and where effort is or was taken towards the goal of equitable benefit. Next slide, please. So this work resulted in a publication late last year, which sought to address several of the objectives set forth in the ICDA white paper. Kristen's kindly just elucidated those. I should note that while objective 2 was to establish best practices for PRSs, The task force certainly recognizes that many groups around the world, such as Primed, ClinGen, and many others, are also working toward best practices, and it's best to closely coordinate and indeed work together on that. Next slide. So as part of responsible use, the task force assessed the potential benefits of PRSs, grounding its findings as firmly as possible in peer-reviewed literature. These included disease prediction, particularly in combination with non-genetic risk factors. Many use cases were looked at here, and the task force felt that the exemplars were cardiovascular disease, where there's a long history of clinical risk prediction and a depth of evidence on PRSs, and breast cancer, where models like Bladicea and tools like CanRisk already incorporate polygenic scores and have CE accreditation in the EU. Next, improving and refining diagnoses, for example, differentiating type 1 versus type 2 diabetes, or more cost-effective use of MRIs for diagnosis of ankylosing spondylitis. Slowing disease progression and recurrence, such as more efficient deployment of costly therapeutics. Improved population screening, for example, using a PRS to reduce unnecessary evaluations of bone density in osteoporosis screening. And prompting risk-reducing behaviors. Now, while the literature is still sparse there is evidence of positive behavioral effects to reduce cardiovascular risk in the Finnish gene risk study. And in obesity, receiving one's genetic risk may alter perceived fullness after food consumption and running endurance during exercise. Next slide. As exemplars of potential PRS benefits, it's been demonstrated that the PRSs for coronary artery disease are both significant predictors of future disease and add independent information to conventional risk factors such as age, sex, cholesterol, blood pressure, and smoking status. So in 2015, Mega et al. Showed that coronary artery disease PRSs can be used to identify a high-risk subgroup who disproportionately benefit from statin usage in terms of relative and absolute risk reduction for incident disease. And more recently, groups from the Odyssey and Fourier trials also found similar for PCSK9 inhibitors, thus demonstrating that PRSs may be used to guide deployment of costly medicines, as well as potentially increase the the efficiency of trials by focusing on groups where medicines have greater efficacy. Next slide. However, for responsible use to occur, the benefits must outweigh the risks. Here, the PRS task force defines several classes of potential risks. There are those from incorrect risk estimates, including well-known ancestry and other demographic biases, which arise from non-representative data, a frequent and perhaps universal inequity of risk prediction tools. I'll give a short plug here to a roadmap to increasing diversity in genomic studies, which was just published in Nature Medicine by task force members Shegun Fatumo, Alicia Martin, and others. There are also risks from incorrect risk estimates, including those which arise from risk communication issues, such as if a healthcare professional doesn't convey the uncertainty, indeed the probabilistic nature of a polygenic score, then the individual may incorrectly believe that their polygenic score means future disease is inevitable, thus sort of playing into the widely held but mistaken beliefs about genetic determinism. This may have knock-on effects, psychosocial effects like anxiety and depression. So there are also societal risks. We've all heard about the commercial use of PRSs for embryo selection. However, the clinical value of using PRSs for embryo selection is likely to be quite limited. And the ethics of parents selecting non-clinical traits or incompletely understood clinical traits in their offspring is ethically dubious. Further, genetic discrimination has played a prominent role in recent human history. Next slide. As just one example, genetic discrimination fueled by outright racism has played an ugly part in past sickle cell disease screening programs. And indeed, leading scientists are not innocent. While he's one among many, Linus Pauling himself publicly advocated that individuals with sickle cell disease, quote, have tattooed on their foreheads symbols for the defective genes they carry. And according to his ribald friend, quote, "It would be better to tattoo the symbols in Braille on their abdomens," end quote. We've also seen genetics commandeered to support current white supremacy movements, such as lactase persistence in white European ancestries and the symbolism of chugging milk. Next slide. In terms of the gaps which remain for responsible use of PRSs, there are clearly still rapid changes to the methods used for their development and evaluation, particularly for standardizing performance across demographies and analytic best practices. For translational aspects, how do we communicate PRSs and the information they hold to healthcare professionals? What are the best practices for return of PRSs to individuals? And how might this vary between cultures and countries? And indeed, if the benefits of PRSs outweigh the risks, how cost effective are they? Finally, significant legal and regulatory gaps remain, including how we classify PRSs. Are they medical devices or clinical decision support tools? To what extent does this depend on the jurisdiction and indeed on the clinical use case? The laws and regulations of each country are quite varied, and because of this, we need to be cognizant that there is substantial costs to PRS approval and compliance with different regulations. Uncertainties and indeed inconsistencies in regulation may themselves exacerbate health inequalities. Next slide. A particular area of focus is trying to understand how to return PRS results to individuals. Many approaches exist, including visualizations using bell curves, percentiles, categorical risks, risk speedometers, and more. There's also a need for careful consideration so that PRS results are not conflated with return of monogenic results, which are more diagnostic in nature. Scale in terms of number of people and number of PRSs that might be returned is also a major complicating factor. There are simply not enough genetic counselors and physicians to employ the standards that are used for monogenic results. Thus, there is a lot of scope for development of new communication materials and methods. Next slide.
Sorry to interrupt, Mike, but just to let you know that you have 1 minute left.
Okay, I'll just breeze right through this. So just in summary, you can sort of see them there. There are many benefits that need to be established, both in the clinic and in how they complement existing clinical practices, including lifestyle regulations. We need to mitigate the risks, in particular, context and cultural competence of PRSs, ancestry biases and societal risk. And there's a series of gaps that need to be closed, in particular when engaging healthcare professionals and how we communicate risk. Next slide. I'll stop there and show again all the wonderful individuals who are part of the PRS task force, and also thank my co-chair, Cristen Willer. Thanks.
Great, thank you, Cristen and Mike, for this very interesting update about their work. And unfortunately, we won't have time for any questions about this talk, and we'll move ahead right away to the pre-recorded talk by the Global Equity Group.
Hi everyone, welcome, and thank you for joining us and for this opportunity to tell you more about the work of the Global Equity Working Group in the ICDA, which I co-lead with Andrés Moreno-Estrada. This work speaks to recommendation 19 from the ICDA white paper, which recognizes that the understanding and treatment of common diseases must be a global activity. So we aim to involve and include scientific researchers from across the globe and take active steps to decrease barriers to equitable participation. And here I've just put our charter around inclusive, fair and ethical practices that support access to and benefits from scientific research for stakeholders. It's worth reminding ourselves why global equity is important. We'll hear a lot, I think, in this meeting and may have already heard about the importance of diversity of research participants, but we also need to support the diversity of researchers. That includes underrepresented groups, minorities, representation of researchers in the global south, and also junior and mid-career researchers whilst practicing gender equity. So a lot of what we're doing revolves around supporting the opportunity to participate and to participate meaningfully. When we first started working together as a group, we talked a lot about what global equity actually looks like. How do we describe it or measure it, and how can we promote it? And we decided it would be helpful for us to build a global equity standard, which would work as a tool to help us understand how global equity can be defined and to help us achieve practical global equity targets. So we've built a framework which describes the key elements of global equity, and we've built a standardized way with metrics to measure those key elements. And we've also defined some practical global equity targets that we can work towards as a group. So the framework is a generalized and constant framework that describes global equity in different ways and with different lenses on global equity. But the metrics that we've developed are context specific. And then the output is a standard which can be used as a tool for self-evaluation and also setting targets to improve global equity. Whilst we're initially using the global equity standard as a tool to evaluate our own organization and our own working groups, we hope that the global equity standard will be used by other stakeholders going forward to support their work towards global equity. So we see the standard as a tool for self-evaluation and target setting in a very practical way that can be used to self-administer monitoring and evaluation and monitor progress. But we also hope that using these metrics and ongoing monitoring and evaluation of global equity using the standard can help to identify possible barriers to achieving global equity and support proposing specific and practical solutions to those barriers. Here I'll briefly take you through the process that we used to develop this standard. So first of all, we wanted to define the global equity framework, and these are the constant themes that I already referred to. We came up with 5 key themes: fairness, inclusiveness, benefit sharing, access, and procedural values, and we'll unpack these themes a little bit further as we go along. In defining each of these themes, we thought about what were the aims that we wish to achieve under each of the themes, and what were the specific areas we were targeting for a kind of shift in practice. And then using these, we were able to develop some specific working group metrics that can actually be measured and that speak to particular activities within the working group. We needed to select a use case in order to prototype our first set of metrics that we put together, and it seemed most appropriate to put together metrics for measuring our global equity achievements and where we need to do some work in our ICDA working groups. The next step was to specify the actual metrics that we were going to use to measure global equity under our different themes. So we had different subgroups working on the metrics for each of these themes, and obviously there was some overlap and we worked through those. We developed quantitative metrics that can be actually measured, and then we also have some qualitative metrics that could be used perhaps in a survey approach. Our metrics have been finalized for this first prototype use case of working groups in a consortium. So I'll take you briefly through at a kind of overview level, but we will soon be publishing this work so that the standard is more widely available to everyone who'd like to give this a go themselves. So for the theme fairness, we had 15 metrics, which included measures of equal opportunity to attend and participate in events and meetings, to access training, and to be able to afford different resources. And we also included some metrics about mechanisms for complaints if there are perceived unfair practices in the working group. In inclusiveness and diversity, we have also 15 metrics, which include measures of inclusivity with respect to different groups. So, for example, with respect to gender, geographical location, representation of traditionally underrepresented groups and ethnicity. And then we also have an assessment of policies, strategies, monitoring and evaluation and implementation plans around improving inclusiveness in the working group. And this diagram just gives you an overview of how we've looked at inclusiveness as also having facets of diversity and representation, and also facets which include policies and frameworks to really formalize how we approach inclusiveness and diversity in the working group. Under the theme of benefit sharing, we have 13 metrics, and these include metrics that assess the policy, the strategies, how we do stakeholder engagement, and also the group's knowledge and beliefs, as well as experience or prior experience with benefit sharing processes. In access, we have the theme of access. We have 9 metrics, which includes the assessment of participant access to participate, access to resources and collaborative opportunities, as well as looking at open access planning and wider accessibility of the outputs we generate as a working group. This figure gives an overview of how we approached thinking about benefit sharing and access in terms of transparency and trust, and about equality of access and benefit sharing. We looked at stakeholders that were internal to the organization, stakeholders that were external to the organization, and then we also considered additional stakeholders that didn't fall specifically into either of these categories. Under the procedural theme, we have 13 metrics, and these look at measurements for processes such as transparent record keeping, the governance structure for the working groups, in terms of references, the documentation, and how we formalize the participation in the working group. And we also looked at decision-making and discussion protocols and attribution and oversight of outputs from the working group. So this is about looking at the structure and how the structure has been put together to ensure that we can uphold global equity and support all the other themes that we've identified. And then finally, for each of these metrics, we proposed targets for our working group. So we had medium-term targets and long-term targets. We selected a 1-year target, a 2-year target, and a 5-year target. And this was about deciding what kind of values would we like to aspire to achieving for each of the metrics to feel comfortable that we're on track to supporting global equity in our working groups. Obviously, this is a very small text, so not to worry about the detail, but this is just to show you that each metric, for example, here we have the theme fairness. Each metric has a line and we have a description, how it's measured, the units, how often we think it should be measured, and then we have targets, 1-year, 2-year, and 5-year targets, all already set up for each of the metrics. And then obviously for fairness, we would have 15 metrics, so there would be 15 rows. And then we have the set of metrics defined like this for each of our themes in the global standard. And just another reminder that each of the themes, fairness, inclusivity, and the other themes, those remain constant in this framework. They provide the framework on which you can add or you can hang your contextually specific metrics. So you may have different metrics depending on what kind of entity you are. You may adapt some of these metrics that we've already put together, or you may have completely new metrics. But what remains constant is the different themes under which you can think about the kind of metrics you'd like to develop to support global equity in your organization or entity. Now that we have these metrics for working groups ready to go, what are the next steps? So first of all, as the ICDA working groups, we will use the standard that we've developed and test out these metrics to measure our baseline global equity in our working groups, so that we can see where we have gaps and we can see what targets we need to set and how far off we are from reaching those targets in order to support global equity as we aspire to do. We also would like to see the standard being used for other stakeholders. Some of the stakeholders we thought about who might find this of use are funders, journals, international collaborations, research consortia and networks, and even individual research groups, and then training programs, and also the biotechnology sector. I'm sure we haven't thought of everyone, but I think there are a lot of end users who might want to use the framework and build some metrics to look at their own global equity and to measure how they're doing in a self-evaluation process and to set some targets and monitor their process towards getting there and reaching those targets. So we hope that this will be more widely usable for any organization that's interested in promoting global equity. So on behalf of the Global Equity Working Group and the ICDA, and these are all our wonderful members from across the globe, we're trying to be very diverse with members from 6 continents, more than 10 nationalities, and a good gender balance. So from all of us, thank you very much for your interest in our work. And we're looking forward to feedback and hopefully interactions, further interactions around the global equity standard in the future. So thank you.
Welcome everyone back to the live session, and thank you, Andrés and Nicki, for this wonderful presentation about the work done by the Global Equity Working Group. So now we have 8 minutes to discuss and answer questions by our two latest panelists who are here. But also the previous panelists are also in the room. So I encourage everyone to ask questions to either the later talk or even some missing questions that you may have in your head for the previous ones. So far, I don't see any question in Slido. And I'll also be checking the Slack, so you can also use Slack to ask your questions. And I see some discussion that has been going on there. So please don't be shy, go ahead and ask your questions. And in the meantime, I will ask Andrés and Nicki whether they can tell us a little bit about what is your impression about the work done by the working group in terms of applying what it's aiming to in terms of diversity and do you feel that the work by the working group is pretty much done, or does it need to grow, continue, and follow and search for more for other goals? So either of you could answer this question.
Well,
go ahead, Andrés.
It's definitely not done, but I think both of us have answers with respect to that. Nicki, do you want to go first? I'm happy to complement that.
No, you go ahead, Andrés, and I'll follow.
Perfect. So, no, definitely the work presented today is pretty much just the very starting point of what this working group can do, and the aims that we have are pretty ambitious. I mean, you saw already. We thought the first step is just to at least diagnose and have a look or a measure of how would global equity would look like, and how can we have an assessment tool for that. But I think once that is achieved, number one, that will become already a tool that can be replicated and used by many other organizations, as Nicki was pointing out. We want to use our own ICDA structure and working groups to have a first grasp of how that is looking internally, but also many other organizations have also expressed interest in also applying this internally, and that will give us a good baseline to work towards in the future, and hopefully improve that not only within consortia and collaborations, but also in other bodies that are really supporting science in many other arenas, like journals, like funders, and many others. But beyond that, I think the global equity, once the whole network is formed, has a lot of potential to do other supporting activities, like, for example, training, which is something that we see as a very important component of also being global. We keep talking about this imbalance about genomic databases and data or knowledge being generated in different regions. But I think the baseline of that is training capacity for all the other trainees and researchers involved in any phase. So that also is another big area of growth that we are planning to also apply as a working group, and pretty much it's also by no means absolutely comprehensive. We're always accepting suggestions and views of what else this group can implement, and I think it's a good opportunity to have this within ICDA, and I'm sure also many other ideas. Nicki, if you want to also complement on that.
Thank you. Yeah, I agree with Andrés. I think we just started. And I think certainly what I've seen in the kind of shift in conversations is that we start off almost like with a topic that global equity and diversity are things that we must pursue, but we don't really know what that means. And then the next step is to start to define what we mean by that. And I think that's where we are now, is looking at what we actually mean by this and starting a kind of operational approach. So we've gone from the concept and we're operationalizing our conversations. So I think that's the point we've reached now. And the next step is to actually do the doing and to make the changes. So the work we're doing now is really flagging the work we need to really get going on. So I think there's a huge scope for us to go from starting the conversation and getting people thinking and talking about where we are in our sort of global equity approaches, and then actually to get to the doing that makes a substantial shift to the way things are operating. Yeah.
Okay, great, thanks. So we have a few questions now in Slido. Anonymous asked Mark Daly during his plenary about what measures he felt was appropriate to increase global diversity and representation in an equitable way. What is this panel thought on that? What are your thoughts about that? What are the appropriate measures?
So if I can just jump in, I think, what I've just mentioned as well is that I think our measures are to try and find some pragmatic and practical ways to move forward and to help the community move forward in terms of supporting global diversity and representation. So that's a large part of why we went for building this very practical framework and very applied standard, is to try and make it possible for it to be easier to pursue global equity standards. Andrés, I don't know if you'd like to add to that further.
Yeah, definitely, it's a good point to discuss and a question that I think needs several angles to evaluate, right? Because we have been discussing this imbalance maybe from the database point of view many times, but not so many times from the composition of those teams and those researchers producing those databases, or the knowledge in general in biomedical research in general. So as a very first step, like we said, this measure will be useful to have a view or an assessment of that, but also to work towards improving that imbalance. And I think that composition hopefully will get to a point in where we can level up a little bit more, not only the representation of the diversity we want to reach, but also the researchers and the local knowledge of people that are more knowledgeable about those populations, to also give back to the populations involved in terms of local capacity building. So I think it all goes about making local efforts to really decisively support locally the research, and not only grow the diversity by itself in the current databases. I think Helen Parkinson, for example, earlier was already acknowledging that, well, there's a lot of new data from same cohorts that are already in there. And there's this will increase the complexity of the data originated by mechanisms, and the other levels of data will increase that bias, in the sense that those that are already advanced will be more likely to produce even more data. And I think we have to distribute that to other types of data from other regions that are more diverse. And I think the only way to do that is to really support locally big programs like it has happened in Africa, for example, with H3 Africa. As you may know, and we will be presenting later on Friday, we have another regional initiative within Latin America, which is called Latin Genome, which is also stemming from ICDA as a regional effort. And I think that's the way to go in terms of joining forces locally to try to balance this diversity and equity.
Thank you, Andrés. And we ran out of time, but I didn't want to finish without one question for the PRS group, which is the most voted one. Now, will multi-omics data significantly improve the accuracy and the biology understanding of PRS? Underline the answer.
So I'm probably on the side of people who believe that to be the case. So multiomics data obviously can help you sort of narrow down to the actual functional alleles. It is also, in and of itself, we shouldn't sort of view multiomics data as being support to PRSs as well. I think this is also a source of data, a source of predictors. You should be combining, PRSs with, all available data to create the most powerful sort of equitable and useful, clinically useful models that you can. So, kind of an easy question. Yes.
Great. Thank you all. Thank you for staying to the end. And we'll go to the break now, and we'll meet 10 minutes past the hour. Thank you, everyone. Bye.